Today, we are jumping into a real-life scientific detective story. It's a medical case that stumped doctors for years, a mystery that was hidden deep inside a family's DNA, and it was finally solved using some of the most advanced technology you can imagine.
So here's the central puzzle, the question at the heart of this entire story. It's the kind of question that seems to break the basic rules of genetics, right? How can a disorder possibly be inherited if the parents don't seem to have it themselves? Let's start by digging into the case.
So, to solve this thing, we first have to get a handle on the specifics of this family's truly baffling situation.
Okay, so here's the family's pedigree chart. The shaded squares at the bottom there, those are the three sons. And as you can see, their parents are both healthy. And if you look further up the family tree, there was absolutely no history of anything like this on either side. This condition seemed to appear completely out of thin air.
And the condition itself was just devastating. All three brothers shared this very specific and severe set of symptoms. They had severe intellectual disability, a complete absence of spoken language, and symptoms of autism spectrum disorder. And on top of all that, they all developed epilepsy. For almost a decade, the family was living with this incredible challenge without a diagnosis, which you can imagine just made everything that much harder. So, with no family history, and no answers from any standard tests, the researchers had to bring in the big guns. Let's look at the high-tech investigation they launched to crack this case wide open. They used a technology called Whole Genome Sequencing, or WGS. Now, the best way to think about this is that it's like reading every single letter of every single word in a person's entire genetic instruction book. It's an unbelievably powerful tool that lets scientists hunt for any typos, no matter how tiny they are. So the mission was crystal clear. These scientific detectives were hunting for one single, shared genetic typo, one tiny error somewhere in the 3 billion letters of DNA that all three brothers had in common, an error that could finally explain everything. Now, as you can imagine, sequencing 3 entire genomes creates an absolute mountain of data. The real detective work wasn't just getting the information, it was sifting through all of it to find the one single clue that actually mattered.
And I'm not kidding when I say a mountain of data. In each one of the brothers, the initial scan found more than 4 million genetic variants. That is, differences from a standard reference genome. Finding the one typo causing the problem in all of that? That's like trying to find one specific needle in 3 gigantic haystacks. Check out how they did this, though. Their filtering process was brilliant. They start with over 4 million potential culprits in each brother. Okay, first filter. They only keep the variants that all 3 brothers share. That still leaves them with over 3 million possibilities. So next, they zoom in, looking only at variants inside 182 genes that are already known to be linked to autism. That gets the list down to 53,000. And for the final step, they search for one very specific clue, a new high-impact mutation. And just like that, out of millions, they were left with just one. I mean, think about that. From over 4 million possibilities, they narrowed it down to a single prime suspect. The scale of that is just incredible. The high-tech search had absolutely paid off. The team had their smoking gun.
So after all that work, what was it? What was this single genetic clue? Let's take a look at the culprit that was hiding in their DNA the entire time.
The answer was unbelievably small. A single letter C was deleted from a gene called SHANK3. Now, this gene is known to be super important for brain function, especially for how our brain cells, our neurons, communicate with each other. And this one tiny deletion, it was the cause of everything. And this slide right here shows you just how devastating that one tiny error really is. On the left side, that's the raw data, and it's pointing to exactly where that C is missing in the brother's DNA. Now on the right, you can see what actually happens because of it. Think of the gene like a sentence in an instruction book. When you delete one letter, it causes what's called a frame shift, which basically scrambles the rest of the sentence. The cell can't read the instructions anymore, and it stops building the protein way too early. The result is a short, useless protein that just can't do its critical job in the brain. So, case closed, right? Well, not quite. Just when the mystery seemed completely solved, the detectives found one last piece of evidence that just didn't fit. A final twist that changed everything. To be sure, the scientists sequenced the parents' DNA from blood samples, and what they found was just wild. The mutation wasn't there. You can see it in the data here on this slide. The spot where the deletion should be is perfectly normal in both the mom and the dad. The genetic typo was only in the sons.
Which brings us right back to our big puzzle, but now it's even more confusing. We know what the mutation is, but if it's not in the parents' blood, how on earth did all 3 of their kids end up with it? This seemed totally impossible.
Well, the answer lies in this really rare but fascinating phenomenon called germline mosaicism. Here's what that means. A genetic mutation is present in a parent's reproductive cells, their sperm or eggs, but it's not in the other cells of their body, like the blood cells they tested. So one of the parents was carrying the mutation in a way that was completely hidden from a normal blood test, but they could still pass it on to their children.
And just like that, the final piece of the puzzle clicked into place. So let's look at what this incredible discovery meant, not just for the family, but for the future of genetic science.
The most important impact, of course, was for the family. As the researchers themselves put it, this discovery helped alleviate almost a decade of anxiety. They finally had a definitive answer. They finally knew what they were dealing with, and that long, painful search for a diagnosis was finally over.
So, what are the big takeaways from this incredible story? Well, first, it gave a family a life-changing diagnosis after nearly 10 years of not knowing. Second, it was powerful proof of just how critical the SHANK3 gene is in severe forms of autism. And third, it was a landmark case that showed how whole genome sequencing is revolutionizing our ability to solve these deeply personal medical mysteries.
And that leaves us with a final really exciting thought. This was just one family and one genetic clue. Now that we have the technology to read our entire genetic instruction book letter by letter, what other mysteries are out there waiting to be solved? What other answers are hidden in our DNA, just waiting for the right detective to come along and find them?